I will invite Ulrich Luhmann to the podium. Ulrich is the biomarker experimental medicine leader for ophthalmology and rare diseases at Roche, and he'll talk about lessons from visual function testing in clinical trials and natural history studies in age-related macular degeneration and diabetic retinopathy.
Yeah, thank you. I'm very happy to share some of our insights in that space in this fantastic meeting. And I try to start out with reminding at least myself of what I'm here for. And maybe we can rally around that support a little bit. So I'm here focusing on the early disease stage in this setting, so the non-proliferative stages. And we are aiming to understand the heterogeneity clinically of that, to understand also the treatment populations which are really in need to be treated in these kind of earlier stages, because they are close to vision-threatening events or to conversion rates to later and with high chance to get clinically meaningful change there. What are the endpoint candidates we want to look at, and how they are meaningful for patients? And by that, we will try to achieve to be able to open up that space for drug development, to get new treatments in that space, utilizing hopefully endpoints beyond DRSS. So I start out with what are the challenges, and I don't think I need to spend much time on it because I think many of them have been mentioned. But in principle, what do we always think about? Who do we want to treat? And we inherently can't get rid of the fact that we have here slow progressing disease. So now our capabilities to run trials in that space are limited in terms of financial and time. So that's one of the key questions. So inherently, we need to run long and large trials, but we face the fact, do we find the right patients on current classification? We have currently high screen failure rates faced by that, by these unclear or variable definitions we use. How do we measure it? And the great news is we have a primary endpoint, that's DRSS. It's predictive, we know that's working, but with all the feasibility challenges, it increases on the variability plus minus one step, requirements to show drug response over existing proof of concepts, anti-VEGFs, for example. We increase further trial burden on that. And regulators request supportive evidence for patient relevance, which also means we put patients on a burden on our patients and the sites to execute that trial, which makes it less attractive to be part of that. So I summarize it and want to really highlight it very briefly. We want to define populations. We want to have that done in a kind of new updated grading system, not revolutionizing what we have because it works, but we want to enable, hopefully, additional information, all of that. And maybe by simply enabling tools to implement what we have more efficiently in trials is also a way to go, I think. We need new endpoints beyond the current ones. And we need to certainly follow a certain step systematic approach how to validate them. And we need to understand the patient relevance. And I want to not mention the unpopular pace. We are also interested in how payers and health economic systems are seeing that. What's the value of what this new treatment in that space may provide? And we need to also consider that because it makes us invest in that space. If it doesn't add any value for the health economy, there's basically no real benefit to developing a new drug in there. I want to really highlight, and so how do we do that? I think we mentioned consortia earlier, and I have to say I'm involved in one of that, in another similar way. It's MACUSTAR, it's a European Innovative Medicine Initiative, where pharma and academic consortia work very effectively together to run such a trial, where we consider actually cross-sectionally and longitudinal data readouts for candidate endpoints, and then take it forward for a 3-year longitudinal trial. The challenge is and remains, do we see enough events to generate that data, linking it up to the vision-threatening end stages? And do we have a clear definition what we powered against in terms of the conversion? I want to also highlight one specific thing from the functional part of that. This is one part where we have really taken the effort, put 8 functional vision tests, 5 vision tests and 2 PROs into that, 1 PRO in that trial. And we found that if we look at that, it's quite a complex thing, but you can see we have all the vision tests here, and we have a complex pattern. We define a 5% threshold on extreme values of asking, is that patient affected by that measure? And we asked that question, how many patients are affected of at least one of the measures we have looked at in an extreme case? So we have here example, 15 patients show dark adaptation problems. But we have also one patient which shows dark adaptation problems, low luminance problems, contrast vision problems, Morpheus acuity problems, simply highlighting the fact that our current classification is not looking at function, but looking at structure, and it's not really well related to function. Can I share something for you, which is probably novel, for the DR space? We have done the effort in a phase 2 setting in Roche, because we are really interested in understanding where we are in the NPDR space. Here, we actually have done something very similar. We actually went through the effort to look at all the different types, low luminance, contrast vision included, and asking the question the same: how heterogeneous in function is the NPDR population defined by 47 and 53 based on inclusion-exclusion criteria? And we see something very similar. If you ask the question, at least one affected extreme case, 70% of patients suffer some degree of functional deficit. The question remains, is it meaningful to the patient? Because none of that leads to 15-letter difference, right? BCVA 20/40 or better. They are not BCVA impaired. However, it's interesting to consider that there is some data, and that self-reported vision-related functional burden questionnaires put out, not regularly accepted, but in these internal efforts by Genentech colleagues, there were findings that severe NPDR have self-reported similar vision-related functional burden in their daily life as PDR. I found it very interesting, and I hypothesize now, we need to understand who are the guys above the green line. Who are those suffering? Are they the one from one particular function deficit, or are they maybe the one which suffers from 4 or 5 or 2, right? Are these then the meaningful ones we should look at? And how do we do that clinically? Is that one assessment, or do we really need to go painfully through 5 of them? So key lessons learned, right? It's feasible to do that, right? We can detect these heterogeneous functional deficiencies in a trial. It comes with a huge effort on clinical side and patient's burden and operational challenges. Can we find those? And how do we learn about that? And the question is regulatory. Can we utilize that data initially maybe as supporting evidence beyond DSS? Or the key learning is actually, and our current staging doesn't allow us to find those patients. So we need to find those patients and understand whether this is prospectively validated or not. And one key hurdle to that is operational. Those functional tests are not very well used. They are commonly not used in clinical practice. They are not distributed in the retina community very well. And so I basically want to motivate with that data our retinal colleagues to really think about function as well. Thank you.
Thank you so much, Ulrich. We have time for maybe one question.
Thanks for that presentation, but you teased us with showing the patients that did very poorly on the patient-reported outcomes. What was it related to? Was it multiple tests or a single test? Do you have results on that, or?
I'm sorry, the visual report outcome, unfortunately, that's not this. Oh, the visual outcome data in my phase 2 trial, I only have the baseline cross-sectional data because the trial is still running. So I'm really looking forward to what is the longitudinal data looking like. We will only know that in about 9 months, unfortunately. A long trial, which took a long time to complete. It's ongoing.
Thank you so much.